Hi class, how you doing? Sorry I can't be with you today. I thought I'd get a start on the chapter 17 notes with you since we have limited time and we got to make use of it. I'm only going to probably talk for about 20 minutes today, and then I'll let you do some review, maybe get started on the homework, and you can possibly take the quiz if you have some time today in class. Chapter 17 is rather large, covers a lot of different topics. The basic concept is the central dogma, which discusses how DNA is converted or DNA is used as the instructions to build proteins. So that's basically what I'm going to start covering today with you. And I will review what you have questions when I come back on Thursday. Please, feel free to get started on reading chapter 17. You will need to start early because it's a lot to this chapter. So let's get started. We'll start with just the basic idea that right here at the bottom of the note, that gene expression is really going to describe how exactly DNA directs the making of protein synthesis. We're going to cover this in two steps when we talk about transcription and translation. And then as we go throughout this topic, you'll see exactly how each of those steps occurs. Um, so yeah, the first chapter in the uh, central dogma topic is on history, and we're actually going to not spend too much time on history, just because there's a lot to get to. So feel free to skip this chapter as we get started on looking at gene expression. So, what to know as we get started here? Uh, as we think about and talk about scientists that started researching how genes are instructions for proteins, there was a few sayings that went around, and one of those sayings or one of those conclusions that came about as research developed was that for every gene, there must be a singular protein that exists in the body. And to some extent, that is true. However, um, we basically revised that hypothesis and conclusion as research has gone on, because we understand now that proteins are not just made up of one gene. And so basically, when you think about how genes are used as instructions to build proteins, we have to remember that proteins are multiple polypeptides that are put together to build one protein. So the scientists that basically came to this conclusion studied how genes related to proteins and determined that for every one gene you have, you basically have one polypeptide. It is common to refer to polypeptides as proteins, but that technically is not correct. You want to make sure you understand that multiple polypeptides have to come together to build a protein. I should also say that this statement here, one gene equaling one polypeptide, is also not always the case. Some genes can be rearranged in between the DNA and the protein outcome, which can elicit multiple types of polypeptides from one gene. So it is still unclear today, even though we know we have about 22 to 23,000 genes in our cells, it is still unclear as to how many polypeptides those genes direct to make. So for every gene, it is still a little uncertain as to how many polypeptides we get out of every gene, because it is very much based on each particular gene. Okay, as we talk about today's topic, we're thinking about how our genes turned into proteins. So at the conclusion of this basic intro here, you can basically determine that genes direct the making of polypeptides, and for every one gene, multiple polypeptides are possible in a cell. What are the steps that it takes to actually build a polypeptide? Those two basic steps are right here, transcription and translation. Transcription is the process of turning DNA into RNA. RNA, like it says up here at the top, is the bridge, the middle molecule between genes and proteins. While transcription focuses on making mRNA, and mRNA is the code that is used to actually create a protein, there are multiple RNA molecules that are used to make this possible. So it is correct to say that RNA being the bridge between genes and proteins is true, even though it looks like messenger RNA is the only RNA molecule at work here. There are many, many types of RNA that help make or direct the making of proteins. During transcription, like I said, DNA, which is in your nucleus, is transcribed and basically transcribed and written into a new molecule called RNA. And for most of us, we think of messenger RNA when we talk about this. Messenger RNA then holds a temporary message of DNA based on what the template code basically said or had in it, embedded in it. And then that messenger RNA is used to make a protein. Ribosomes, which remember we talked about in class, are our chefs, read the messenger RNA, assemble the ingredients, which are your amino acids, and, and bond them together in the correct order, which allows for the making of a polypeptide. So ribosomes, again, will read messenger RNA and assemble a polypeptide by bonding amino acids in the correct order. That process is called translation. In eukaryotes, this actually is a little more complicated than in prokaryotes. Prokaryotes, transcription and translation can happen at the same time, but in eukaryotes, because there's a nucleus and there's extra genetic material in eukaryotic cells, extra steps have to take place in order for DNA to turn into protein. So maybe I'll come back to this picture here in a second, okay? Basically, the central dogma, which we've mentioned before, is the concept of turning DNA into proteins through the conversion of, or through the transition to RNA. This is that basic model. Here's what it kind of looks like in bacterial cells or prokaryotes. DNA is transcribed into RNA, and then RNA is translated into a polypeptide. In bacterial cells, because there's no membrane separating the DNA from the rest of the cell, this process can happen all these processes can happen simultaneously. In eukaryotes, DNA is in a nucleus, so it's transcribed into RNA inside of a nucleus. And then as soon as the RNA or messenger RNA leaves the nucleus, it can be translated into a polypeptide. There's also this extra step called RNA processing, which we will talk about. Eukaryotic cells possess coding sequences that do not or are not required for the assembly of a polypeptide, and so they have to be removed. That extra step involves some extra proteins and molecules to get that stuff completed. So how are instructions for the assembly of proteins encoded in DNA? Well, they are basically encoded in those 4 nucleotide bases. We're talking about A, T, C, and G. Each one of those nucleotides in order directs the assembly of the making of proteins. Amino acids, because there are 20 different amino acids and only 4 nucleotides, one amino acid cannot code or be coded for by a single nucleotide. Multiple nucleotide bases have to have to be read in chunks to basically direct the making of an amino acid or the assembly of a polypeptide. So how is that done? Well, the gene is read in a triplet code. So nucleotide sequences are read in groups of 3 nucleotides at a time, and every 3 nucleotides corresponds to one amino acid that is one link in the polypeptide chain. What that looks like here is, here would be my template strand for you thinking about DNA and that the replication activity we just completed. The template strand is essentially the gene. Up here we have our gene. In order to copy that, transcription will make a complementary mRNA sequence that does not directly match the template, but is complementary to it. The complement is read in sections of 3. Each section of 3 is called a codon, and each codon codes for one specific amino acid. Each amino acid in the correct order will allow the polypeptide to fold up in its correct shape, which will give it functionality in the cell or wherever it ends up in the organism. Okay? Template strands. Template strands are essentially genes. Those genes are copied into mRNA, read as codons, and those direct the making of amino acid chains or polypeptides. What does the codon framework look like? Well, there are actually 64 codons that direct the making of polypeptides. Let's see if I can find that codon chart. Sorry, don't know why that isn't in here right away. We'll go back to the beginning, but I think it's pretty important that you actually see this triplet code. The codon chart you're looking at in front of you is a redundant code because multiple triplet codes can code for the exact same amino acid. Right here on the right-hand side of each column, you see an amino acid. So this is proline. Pro is a shorthand for proline. Down here is threonine, alanine, valine, isoleucine, leucine, serine, and so on. Each one of these amino acids is coded for by triplet code. The triplet code is on the left-hand side of the column. Nucleotides on an RNA molecule that reads CCU would code for proline every single time. As long as the code isn't mutated, proline will show up in every single protein or polypeptide where this coding sequence is involved. Okay. All right. What is so important about this is actually quite amazing, and it shouldn't go understated. The genetic code that's present in our cells is universal, which means that bacteria use the exact same codon chart to create polypeptides as complex animals like us use. What that essentially means is that we can insert or um, work in the lab on organisms like bacteria and inject genes into their cells that they can read and make proteins out of. An amazing discovery which has helped lead to so many other ones in medicine. Um, Here are a few very interesting examples of this technology at work. Scientists have been able to engineer tobacco plants that will glow when they need watering. How is that possible? The gene from marine jellyfish can be used and inserted into embryonic plants that then grow with this gene inside of them and are activated when needing water. There are other types of organisms that have been genetically modified using this technology, such as pigs. Pigs have been able to express, um, actually, you know what, I mixed up here the jellyfish and firefly genes. Sorry, guys. Firefly genes are expressed in these tobacco plants when they need watering. It's the jellyfish gene that's expressed in pigs when they have actually received the gene from uh, that's been basically genetically inserted into their cells. So what's happening in this image is that you're seeing an adult pig, or actually probably a juvenile pig, um, that has been genetically modified in the lab as a single embryo. Scientists try and inject multiple genes into a pig that will allow them to either chemically or really naturally make milk with medicine in it. Okay, in order to be able to easily tell which pigs have been genetically modified, which haven't, or which embryos took the gene and which didn't, they usually attach on to those genes a jellyfish gene that glows. That way, if the pig has received the medicine or the gene that will allow them to make medicine, that pig will also glow. So it is easily recognizable as a pig that has been genetically altered. Okay, this has happened in a lot of other instances. There are plants that have been genetically altered with genes from Arctic fish so they can withstand cold winters. Um, there are multiple different types of farm animals that have been engineered to produce medicine in their milk. Quite amazing research. One other one that comes to mind are bacterial cells that have been genetically engineered to eat and process oil from oil spills. Quite amazing technology, and I'm sure there are more uses for it besides the one I've listed, and even the ones that have been discovered today, since today, up until today. All right. Lastly here, before we go, because we're approaching here 20 minutes, we can take a quick look at transcription and get started on this topic. Transcription is the first stage. This is the one you're going to build out first in the model kits, so you can actually see how transcription occurs. Um, how does this process get started? Well, the enzyme you're going to want to focus on here is RNA polymerase. RNA polymerase does all the work, and it actually has the same name or a very similar name to an enzyme from last chapter. When we talked about DNA polymerase and DNA replication, the polymerase enzyme is an enzyme that makes a polymer of whatever the enzyme's attached to, right? So DNA polymerase is an enzyme that makes a polymer of DNA. This enzyme makes a polymer of RNA. It does that by reading the DNA strand and assembling RNA nucleotides complementary to the DNA that is being read. What what you have to remember here is that RNA actually includes uracil in its composition instead of thymine. So RNA is going to look a little bit different than DNA, but if you can substitute uracil for thymine, essentially the molecule is the same. Okay, there are a few regions of DNA that are important when we talk about transcription. And one of those is the promoter, which we'll start with, and the other's the terminator, which we'll end with. Okay, here's a brief overview of this, and then maybe we can go into some more detail Thursday when I see you. Okay, so let's just finish off looking at this picture. The promoter region of every gene is going to be a region that is activated by either the cell itself or the environment the cell is in. Promoters have tags on them which attract RNA polymerase to the gene, signaling that it needs to be copied. Once RNA polymerase lands on the promoter, because it has been turned on, RNA polymerase will unwind the DNA, read the strand, and make a very quick complement of the gene being read. The DNA that's being read is only on one side of the molecule, so the template strand is the gene. The non-template strand has no purpose other than to serve as like a negative of the gene. If there's ever a mistake in copying the gene or replicating it, it can be fixed using the code on the non-template strand. Okay. Once the transcript has started being made, RNA polymerase will move down the molecule until it reaches the termination zone. The termination zone is typically an area filled with a lot of thymines or a lot of the same type of nucleotide that signals to the RNA polymerase enzyme to jump off the strand, and you have your completed RNA transcript. In eukaryotes, there's still a little work to do because remember, we have to cut some of this RNA transcript out. But in bacteria, this molecule is ready to go and can now be made into or read and processed into a polypeptide. When I see you on Thursday, we'll talk about these three stages of transcription. Why don't you take a look at the coloring activity I gave you or some other um, material in the book to try and get a handle on the overview of this process. When we talk on Thursday, we'll try and get through transcription so that maybe you can even make that stop motion video on Friday. I hope you have a great day. I'm sorry I can't be there with you today, and and I'll see you on Thursday. Bye.